Hi everybody, and thanks for taking the time to come today for our briefing today. We are hosted, of course, by the Institute for Gene Therapies, or IGT, and the Alliance for Regenerative Medicine, or ARM. I'm Erick Paulsen. I'm the chairman of IGT, and I'll help moderate today's discussion, which is going to be really interesting and exciting. Of course, we are in a year now that has seen many changes happening across the scientific field of gene therapy, and we're in a new environment, and I think all of us at IGT and ARM in particular believe that we are at a crucial moment to make sure we are educating policymakers to be a good resource, answer questions about the value of cell and gene therapy and the essential need to make sure that our healthcare policy is keeping up with the science and where we're headed. So I also want to acknowledge that we have several of IGT's Corporate Advisory Council members in the audience today, representatives of the manufacturing side of the equation. We have patients, of course, and advocates of several rare disease communities who are with us today. It's also great to see so many Hill staffers here who are part of the policy team as well, because this is an event that gives us an opportunity to bring together all of these different stakeholders to talk about this topic. I also want to recognize Donna Christensen, who is the chair of our Scientific Academic Medical Advisory Council at IGT as well, and one of my former colleagues in Congress. So with that, why don't we dive right into it, and we'll let Erick Cischke, who is the Vice President of Government Affairs at ARM, introduce our panelists, and we'll go from there.
Yeah, thanks, Erick, and thank you everyone for being here today. I also just want to acknowledge that we have multiple members of the ARM Patient Advisory Group and a number of other ARM members here. And thank you again to our panelists for taking time out of your busy schedule. So I'll just quickly jump ahead and introduce our panelists. So I have Dr. Kiran Musunuru, Professor of Cardiovascular Medicine and Genetics at the Perelman School of Medicine at the University of Pennsylvania. You may know Kiran because he and his colleagues have been in the news lately. He's one of the clinicians that was treating baby KJ with the first personalized CRISPR gene editing. So we're really excited to have you. We know you've been on this circuit and very busy. I think everyone is closely following and celebrating that amazing achievement. I have Annie Virginie Egeman, who sits on the ARM board. She is also the Chief Regulatory Officer at Tessera, leading all of their regulatory work with the FDA. And she previously worked at Bluebird Bio, where she helped oversee the FDA approval of the sickle cell gene therapy. And I have Nate Plassman, a father whose son has Duchenne Muscular Dystrophy and received a gene therapy. And I also have Maria Kafalis, who's the co-founder of the Calliope Joy Foundation, Cure MLD, and is just a tremendous advocate, and we're so pleased to have you. So before I jump in, I just wanted to take a moment to acknowledge, as Erick did, that this is really a unique and exciting time in cell and gene therapy, but it's also a unique and exciting time here in Washington, D.C., with lots of conversations happening about biotech, about medicine, about scientific innovation. I think a lot of folks know 2 weeks ago was a really exciting time. The Department of Health and Human Services and the FDA held the first ever roundtable on cell and gene therapy. Folks from the Alliance for Regenerative Medicine were there, and a number of patient advocates. And we were really excited to see that we had FDA Commissioner McCary, but we also had HHS Secretary Kennedy, we had CMS Administrator Oz, we had the NIH Director, Bhattacharya, and the head of the Office of Therapeutic Products, Dr. Verdun. I know I just listed a bunch of names, but the point is we had all of these people who are key decision makers together in one room focused on cell and gene therapy pretty early in the administration, and it's clear that it's a priority for them. And it's a bipartisan priority, which these days is somewhat harder to come by. And so building on that, we want to continue the conversation with all of you on the Hill about what we can do together in a bipartisan, nonpartisan manner to bring more of these amazing treatments and cures to patients and families like all of you. So I'll turn it back to you.
That's a perfect setup, actually, for our conversation. Before we get into any policy weeds or deep into that, I do think it would be helpful for all of us to maybe to get a basic understanding about cell and gene therapies, how they work, how they're different than traditional pharmaceuticals or biologics, and why they're so important in today's world. So with that, maybe We have a scientist and doctor tag team that will start us off, but maybe Dr. Musunuru and Avi, talk a little bit about the background or share some experiences about why cell and gene therapies are so unique. Doctor, if you want to start.
So I think you all have some familiarity with cell and gene therapies, but let's start with the basics. There is this tendency to think of cell and gene therapies as medications. Certainly the FDA tends to view them that way, but it's probably better to think of them more as procedures, as interventions, or even surgeries. They're typically one-time therapies that we hope will have a long-lasting, durable, even lifelong benefit. They can take the form of engineered cells that will seek out and destroy tumors deep in the body while leaving healthy tissue alone. They can take the form of therapies that make precise pinpoint changes in genes and proteins that can correct misspellings in the genetic code, and in so doing, heal patients with otherwise incurable diseases. They can help people, they can protect people from the leading causes of death, like heart disease, like cancer, as well as from syndromes that are so rare, they might only affect one person in the world. They don't even have a name, they're that rare. And then everything in between. So as you heard, I'm a cardiologist who focuses on heart disease, although I am also a scientist. But in my capacity as a physician, I refer patients to cardiac catheterization procedures or to triple or quadruple bypass surgery all the time. And none of us really thinks twice about it, even though these procedures are nowhere near 100% safe. We're so used to the idea of these procedures and surgeries, right? And they're not inexpensive. Major surgeries are easily, it can run in the hundreds of thousands of dollars or even $1 million. But our healthcare system reimburses for these surgeries just as one-time charges. There really isn't any controversy about paying for them because we understand that they help patients in a big way. And we all have family members or friends or coworkers who have benefited from procedures and surgeries. Cell and gene therapies have the potential to be as helpful, if not more helpful, than procedures and surgeries. They can do things like put life-threatening cancers into more or less permanent remission. They can treat devastating genetic diseases like sickle cell disease and baby KJ's disease, which is an obscure disease called CPS1 deficiency. And it can make it so that patients can live their whole lives essentially free of those diseases. Now, again, speaking as a cardiologist, they can get at the root cause of heart disease. They can give you lifelong protection against heart disease, push off heart attacks and strokes by decades, increase life expectancy. This is truly transformational stuff. We've never been able to do these kinds of things before. And what's more, these therapies are usually much gentler on the body than major surgeries. They're much more precise. They have fewer risks. So again, these are not medications. They're not things that you would take every day for the rest of your life or every few weeks or every few months. This is something quite different. It's really more like one and done. Now, it's natural to get sticker shock when you hear about a gene therapy costing $1 million or $2 million and how it's going to break the system. But that's only if you're thinking about it as a medication you take over and over again. If you think of it as a procedure or a surgery that we hope will have a lifetime of benefit, that will lead to a longer, healthier life, or to flip it around, that will reduce major suffering, that will keep people out of the hospital, greatly reduce the number of hospitalizations for a patient, the long-term economics do actually make sense. We just need to be more creative about how these cell and gene therapies are reimbursed. And then in the same vein, there's a lot of discussion about how these cell and gene therapies aren't easy to scale. But again, going back to my world again as a cardiologist, nobody talks about how bypass surgeries aren't scalable. If more patients need surgeries, then we train more surgeons and nurses and so forth, and we credential more hospitals so that they have operating rooms and can host the surgery. So what we really need to do going forward is to think about how to better foster the development of cell and gene therapies, how to appropriately reimburse them as procedures, how to train physicians and providers who are well qualified to administer these therapies to patients, and perhaps most importantly, how to make these therapies as accessible to as many patients around the country as possible. And it is without question that strong federal support, whether it's robust funding from the National Institutes of Health or regulatory innovation from the Food and Drug Administration, strong federal support is indispensable if we want to bring new cell and gene therapies to people in this country.
Thank you. When I think of gene therapy, I think of potential cures. And when I started my career in the late 90s in Paris, this was when one of the first clinical trial for gene therapy was initiated, and it was for a disease called XKID, where boys are born without a properly functioning immune system. So they have to live in a bubble. And these 10 patients were treated in this trial. And out of these 10 patients, 9 had a successful outcome from the treatment. And unfortunately, a few years later, 4 of them developed leukemia. And out of these 4 patients who developed leukemia, 3 of them were successfully treated. And today, the first patient who was treated in this trial is now 27 years old, living a normal life. And the patient who had successful treatment are also living a normal life. And this story, to me, highlights the key to gene therapy. It can bring tremendous benefit, but there are some risks related to these therapies. And in my line of work, where I primarily partner with the Food and Drug Administration, this is how we think of developing these therapies. We think of benefit and risk, and we want that balance to be positive. And it takes a long time to develop these therapies. So a lot of what I'm passionate about is how to continue innovation in the field of regulation. And these regulations should follow the innovation of science. So that is what is really unique about gene therapy is, as Dr. Musunuru just explained, the science is cutting edge. It's evolving every day. So how does the regulatory world evolve along that? It should follow the science. And I look forward to partnering with the new administration in making some of the great ideas that have been shared lately come into practice to really help the development and accelerate access to patients.
Perfect. Well, maybe Dr. Musunuru, you can talk a little bit more about baby KJ and share that story, but also talk about, you referenced strong federal support is essential. Talk about how NIH and FDA helped made that possible in the perspective of when you're discovering, developing, and deploying new gene therapy treatments.
Yeah, absolutely. So I think many of you are familiar with baby KJ. You might have seen the cute, very, very adorable little boy in the headlines. I'm just along for the ride. Baby KJ is the one who's famous, right? But last August, KJ was born with a devastating genetic condition called CPS1 deficiency at my hospital, the hospital of the University of Pennsylvania. This is an incredibly rare disease. It affects 1 in 1.3 million people, so it really is this 1 in a million type disease. And KJ was missing an enzyme in the liver that's needed to break down protein in the diet into metabolites that can be cleared from the body. We do it all the time. Now, because he didn't have this enzyme, there was a toxin called ammonia, a waste product that's made directly from protein that was building up in his body. And when the ammonia level spikes, it can cause irreversible injury to the brain, it can cause coma, and then even death. And in fact, half of patients with KJ's condition, the CPS1 deficiency, they die in early infancy. Now, KJ became very sick within 48 hours of birth. Fortunately, the diagnosis was made by one of my colleagues very quickly. He was able to be transferred to the Children's Hospital Philadelphia CHOP, get life-saving dialysis to clear the ammonia out of his body, but that was only a temporary solution. He was at risk for the ammonia building up again at any time, even with the mildest of stressors, something like a common cold that most of us would just shrug off. It could be life-threatening for him. And in fact, that did happen a few times over the first few months of his life. He'd get a cold or he'd get a stomach bug, and his ammonia level would shoot up, and then we'd all get scared and be watching him very, very closely and try to do heroic things to manage the situation. And because of this, because of the fragility of his situation, he never went home. He was in the hospital continuously from the time he was born, staying at the Children's Hospital Philadelphia at CHOP for many months. He was not leaving. His only hope was to eventually get a liver transplant, which would fix the problem of having this broken enzyme or this missing enzyme, but putting in a new liver that had the working enzyme. But that's a heroic thing to do. It has its risks, and it's certainly not safe to do that type of transplant until typically a patient is over a year old. So he was just waiting in the hospital. And all of us were praying on a day-to-day basis that his ammonia levels wouldn't spike and that he wouldn't suffer any permanent harm. And we've certainly seen plenty of patients where that does happen and they deteriorate over time and sometimes don't even make it to transplant. We couldn't give him a normal diet because the protein would get broken down into ammonia, so we had to heavily restrict his diet. And no surprise at all, he was very small and underweight for his age. So collaborating with my close colleague, Dr. Rebecca Ahrens-Nicholas, at CHOP, who's a pediatrician and a scientist who focuses on treating diseases like CPS1 deficiency caused by broken or missing enzymes in the liver. We've been working on figuring out how to use a very new gene editing technology called CRISPR, which I'm sure many of you are familiar with, to help patients like KJ. We can use gene editing to correct misspellings in the genetic code that cause diseases like CPS1 deficiency. But in this case, there were 2 challenges we had to overcome. First, KJ's disease is extremely rare. It's that 1 in a million. And even more than that, almost all patients with that kind of disease have their own unique misspellings. It's not the same misspelling you see over and over again. So that meant we had to develop a therapy that would specifically correct KJ's misspelling to fix the specific flaw in his broken enzyme and get it working again. And so it would have to be a truly bespoke therapy made just for KJ. The second challenge is that the clock was ticking. We didn't have the luxury of spending years to develop and perfect the therapy. Even 1 year would have been too long. We realized we only had several months to make the therapy if we were to help KJ in time to make a real difference. And so in both respects, this personalization as well as the need for speed, it goes without saying, nothing like that had ever been done before. But Dr. Ahrens-Nicholas and I had wanted to be able to help desperately sick patients like KJ, and so we'd already been working on streamlining the drug development process. We were able to do this only because we were getting funding from the National Institutes of Health, specifically the NIH's Somatic Cell Genome Editing Program. Nobody else was going to give us funding to work on these ultra-rare diseases. And then last summer, when KJ was born, we were ready. Several days after he was born, we got genetic testing results back. We knew exactly which misspelling we needed to fix. And then over the next 6 months, working with various partners in academia and industry, and supported by special supplementing funding from the NIH, specifically for this purpose, we were able to make the bespoke therapy just for KJ. Now, crucial to the success here was our collaboration with the Food and Drug Administration, the FDA. We engaged with them early. They fully understood what we were trying to do, KJ's desperate condition, the need to move quickly, and they committed to work with us to streamline the regulatory process, not let perfect be the enemy of good. And once we had the drug made and did work in the laboratory to ensure that it was reasonably safe and effective, we submitted our application to the FDA. They asked us a lot of questions over the next few days, but then just 1 week after we submitted the application, they approved it, and were able to give KJ the treatment in late February. The treatment involved simply using an IV, infusing the therapy over about 2 hours. He was asleep the whole time. We were very nervous and excited at the same time, but he was asleep and didn't even know what was happening. That was probably a good thing. And the therapy, once you do it, it goes into the bloodstream, it goes straight to the liver, and it makes the fix, and the liver is where the enzyme needs to be fixed. The misspelling, corrected to the right spelling. And so something happened, something very remarkable happened right after we treated KJ. His ammonia levels stayed normal, and in fact, they have remained normal since that day that he got the first treatment, despite having several colds and stomach bugs, which before the treatment was causing ammonia levels to spike. We didn't see that anymore. And we were able to wean him down on some of his medications. And more importantly, we were able to start giving KJ a normal diet with a full amount of protein. And he just soaked it in. He went through this huge growth spurt. He's been thriving since then. On the day before treatment, he was at the 9th percentile of body weight for his age, so very undersized. He's now at the 45th percentile. He's basically average. He was adorable even before the treatment, but he's even more adorable now. You've seen the pictures. And as many of you I'm aware, 2 weeks ago, he was able to go home with his family for the first time, and he's been doing great since then. So what did we learn from all this? Well, we learned that it really is possible to make bespoke gene editing therapies in a reasonably short amount of time, in time to help devastatingly sick children. If you'd asked me even a few years ago, we'd be where we are now. I'd be like, sounds like science fiction. It still kind of does. I still can't totally wrap my head around it, but we did it. And what we did for KJ, in principle, could be done for patients with a variety of other genetic diseases. It'll take a lot of work, and so my most important message is this. This is not the time to declare victory and take our foot off the pedal. There are many KJs who need our help, and we're in a position to double down, to intensify our efforts to help tens of patients, hundreds of patients, thousands of patients like KJ, and eventually build to the point where it's not science fiction. It's the furthest thing. It's exactly like we think about triple and quadruple bypass surgery. It's so routine, it's standard of care. It's just part of everyday living. We don't even think about it as a miracle anymore because it's that routinely done. Now, it'll take a lot of work to get there, but in order to make this happen, we need to ensure that there's continued strong support from the NIH, from the FDA, and I just want to make one last point. I know I'm talking maybe a little bit too long. Federal funding, public funding was crucial to everything in this case, right? So just the understanding of CPS1 deficiency, this 1 in a million disease, if there hadn't been funding to study the natural history of such diseases, and to get a sense what happens when a patient gets this disease and realize, wow, they're going to be in really bad shape and they're going to have terrible outcomes, we were able to predict from the get-go that KJ was going to face a very difficult course, and that gave us the impetus, the need for speed to try to make this therapy as quickly as possible. It was very predictable. CRISPR itself, believe it or not, the way CRISPR was discovered was because people were studying what makes certain bacteria culture yogurt better than other strains of bacteria. It turns out it's because certain strains were more resistant to viruses floating around that kill bacteria. And when scientists dug into what was protecting certain bacteria, they discovered that bacteria actually have an adaptive immune system, believe it or not, to fight off viruses. And from that, took some years of work and a lot of funding, but from that came this amazing gene editing tool, CRISPR, that's now helping little kids like KJ. So it's not always easy to see the path from A to Z. The funding was critical for, the delivery method, the actual type of therapy, figuring out how to use soap bubbles to carry cargo in a solution that you infuse into the bloodstream and go straight to the liver, then be able to fix enzymes in the liver. And these are just a few examples. All of this would not have been possible, or at least not very easily possible, if it hadn't been staunch support, continuing support from the National Institutes of Health, and a commitment from the federal government. So I'll leave you with that point.
So really cool stuff to not only help KJ, but patients with ultra-rare diseases, rare diseases, and there are so many opportunities, of course, to modernize our healthcare system and the patients and families that stand to benefit from them. Nate and Maria, in particular, are both powerful representatives in terms of their personal stories about how they have benefited and seen the benefits of these therapies. So maybe at the end of the day, the patient journey is kind of critical. You can share a little bit about your patient journey, and Nate, you can start with your son Andrew, for instance.
Yeah, thank you, Chairman. My name's Nate Plassman, and for the first 30 years of life, I simple kid from Granville, Michigan. Michigan shaped like a mitten. I grew up on the west side. I thought you might like that. Yeah. 2010, moved to Chicagoland, and my wife, Sarah, along with our 3 kids, we live about 15 miles straight west of downtown Chicago. I brought some pictures with, but as I was going through them, I thought, oh my goodness, I don't have a current family picture. So I've got some snippets. These are our 3 kids. Grace is 16, going on 26. Part of the reason we don't have a current family picture is because she's always on the go. She likes to hang out with her friends. She's very social. Jackson is our middle child. He is 13. He's a wonderful young man. And then Andrew, our youngest, is 11. And when he was a baby, he was born back in 2014, my wife just noticed that he was kind of floppy. He wasn't very confident on his feet. He never crawled. So Sarah's type A to the max. She knew something was up, and she pursued early intervention through the state of Illinois. And we were really fortunate that she did that, because typically for boys or young men that have Duchenne Muscular Dystrophy, they don't get a diagnosis until they're 5 or 6 years old. And when your life expectancy is anywhere from 16 to 26, each day and each week and each month and each year are really critical, because time is muscle. So with Duchenne Muscular Dystrophy, his body doesn't produce what I like to call the shock-absorbing protein. I'm not a scientist. I'm actually just a simple guy who runs a family trucking company. So correct me if I misspeak or I assert something incorrectly, but his body doesn't produce that shock-absorbing quality. And for anyone that's ridden in like a beat-up taxi or Uber car, you can feel it when a car doesn't have proper shocks. Every bump, every pothole, any bit of uneven pavement is really kind of painful. So in Andrew's situation, his body is not producing dystrophin. So over time, what happens is his muscles fibrose. They harden, and they're really self-destructing because they don't have dystrophin. Dystrophin is the largest gene in the human body, which makes it the most susceptible to deletions, duplications, out-of-frame mutations. So we got his diagnosis on the day after his 2nd birthday, July 2, 2016. And it was a day that really changed the trajectory of our family's life. Because you can't ever be prepared for news like we received. Yeah. So all of a sudden I found this new hobby, and it was being a renegade dad who doubled down and dove in. Or a pain in the neck. Or a pain in the rear end, whatever you want to say. And I'm not a scientist, so I learned a lot really quickly. But we were really fortunate. Andrew was selected to participate in a groundbreaking clinical trial at Nationwide Children's Hospital in Columbus, Ohio. And he was dosed with gene therapy on July 11, 2019. And a couple weeks later, we just knew that he had received the live treatment and not placebo, because he was able to do things that we never thought possible. So, I'm the designated crier, the DC on the panel here. I grew up with 3 sisters, so I'm used to being emotional. But this is Andrew today. He's 11. He'll be 11 on July 1st. And he functions like a relatively normal, healthy young man. Now, granted, when we are out and about, I have to be really cognizant and intentional about like the pace of how we walk, or, we always want to try and park as close to the entry of wherever we're going, because he is compromised. It has arrested the progression of the disease, and we hope in the future that he'll be eligible for maybe a CRISPR technology, or, Lord willing, the commercial company that sponsored his trial will devise a way to redose him so that, over time, the durability of gene therapy potentially wears off a little bit. But if they can figure out redosing, he'd be a great candidate for that. He's a happy, joyful, outgoing little guy. He wants to be a park ranger when he grows up, or a police officer. He's a helper. He has a beautiful spirit, and I'm just really thankful and proud to be his dad.
Well, thank you for sharing not only your advocacy for Andrew, but your advocacy for the new frontier of medicine in this space. And Maria, you are a rock star as well and have a story to tell, so we look forward to hearing from you.
Thank you. So 13 years ago this month, in 2 weeks, my youngest daughter, Calliope, was diagnosed with metachromatic leukodystrophy, an ultra-rare monogenic disorder, one of the thousands of disorders which is a prime target for gene therapy. And she was 2, and she was falling down. She had been always running and stuffing meatballs in her face and kicking her brother in the head, which was one of her favorite things to do. And all of a sudden, she fell down one day. And my husband screamed in fear. Like, we had an early intervention team. We knew something was a little bit off. We thought maybe some sensory issues. Anyway, we walk into the Children's Hospital of Philadelphia, which is sort of the recurring theme today. And I have the great pleasure of working with Dr. Rebecca Ahrens. And I encourage all of you to invite these doctors to your staffs, to have them speak, or better yet, come to CHOP and meet this team in person. Come and visit them. They are an inspiration. They should be heroes with a stamp one day. Anyway, Cal was diagnosed in 2012. And I met a brilliant young neurologist, and I said, I can't believe there's nothing you can do for her. And she said, I promise one day we will cure this disease. It will be 10 to 20 years from now. Well, on the year anniversary of Cal's diagnosis, metachromatic leukodystrophy causes a missing enzyme. You're getting a nice ground rounds training on metabolic disorders, guys. Enzyme deficiency, a missing protein that caused catastrophic damage to the brain and central nervous system. Her brain was so damaged when they did the MRI that you could see the brain retracting from the skull. So Dr. Waldman says, one day we will treat this disease in 10 or 20 years. And I said, promise me that when that happens, Dr. Waldman, I want to meet the baby you get to save. Well, a year later, and this is absolutely true, Science magazine published a report, a science note by a team in Italy. They had successfully treated 3 children with MLD. Their MRIs were absolutely clear. This is an early, very early form of a lentroviral-based ex vivo gene therapy. And I called Dr. Waldman, and I was not happy. I was angry. I said, I thought you said 10 to 20 years, not 1 year. How could you not help Cal? And she said, Cal, it's too late for Cal. And look at the research note. The only way these children were treated was because an older child was identified who was too far gone and too symptomatic, and they found a baby. And at this point, I was pretty distraught, and I was raising money for palliative care because that was the only thing I could do. Cal was dying. My house had been transformed into a hospital in a matter of 90 days. We had given away all her toys and her balls and her soccer equipment. My husband was a soccer coach, and he had looked forward to the day to train her to play soccer. And it had all been given away, and we now had a hospital bed and hospice nurses and feeding tubes and 12 medications, and I became a really good nurse. Anyway, Dr. Waldman says, Maria, I know what you need to do with the money. You need to start getting children to Milan for this treatment. Every time you find a baby, you need to get them on a plane. And that's what I have spent the last 12 years doing. As of today, I have gotten 25 children this treatment from all over the world. In fact, shout out to CHOP, this week we dosed our first baby with MLD. Dr. Waldman, that young doctor who took care of me, is part of that medical team at CHOP. And baby Nicholas flew not from the moon, but he might as well have been from the moon. He was identified in the Yukon Delta in Alaska. It took 24 hours for the family to get to CHOP. And on the 13th anniversary of my daughter's diagnosis, I get to have Dr. Waldman fulfill that promise. I got to meet the mother whose son would be saved. And I can tell you that the only thing that has saved me from the despair of losing a child from the most horrible disease you've never heard of, MLD, imagine ALS and Alzheimer's in a 2-year-old. The pain that these children experience, I won't even speak of it publicly. It is horrifying because their nerves are dying. And when your nerves die, they do not die quietly. And to have in my phone pictures of children from literally England, Australia, New Zealand, Brazil, Italy, that have gotten a chance to grow up and live. And this gene therapy is the most durable, most safe one on the market. The oldest US patient is 15 years old and has no symptoms of MLD. Nothing. If he walked into this room right now, you would have no idea that he should have died by the age of 5.5 and cost the healthcare system millions. MLD children do not die inexpensively. My daughter's care cost $2 million, and she had 1,722 confirmed provider contacts over a decade. The economist policy people can do the math. It is very expensive to not intervene with these children's care. Gene therapy is the future. And I want to quickly point out that the first gene therapy patient was treated at the NIH in 1990. Ashanti De Silva was, and they should have a plaque at the NIH for this, which annoys me that they don't. And she is 40 years old. So, mic drop there. Thank you.
Wonderful. Well, it's okay to applaud. Well, Maria and Nate, thank you very much for sharing your personal experiences, perspectives, and meaningful anecdotes. It's a reminder to all of us about why we do what we do. We're going to dive a little bit deeper now, actually, and some of the work that our federal agencies are doing, and talk a little bit more about how that can help patients and families just like Nate's, just like Maria's. AV, you have extensive regulatory experience interacting with the FDA. Talk maybe a little bit about the positive change that you've seen at the Centers for Biologics and Evaluation, at the Office of Therapeutic Products recently, over the years, and how do you build upon that?
Sure. So perhaps to start with, what people I think need to understand is that for therapies like the one that Nate and Maria described, it takes about 10 years to get to the market today, or it took these drugs between 6 to 10 years to get to patients and to be approved. And if you think of the regulatory process, it's gating in 2 principal milestones. One is to initiate clinical trials, and one is to get approval. And there's a lot of back and forth in between. And I think there has been great progress made by the administration, and I'll name a few things that I think we need to continue. One is related to the approval. There is a pathway that's called accelerated approval that allows being approved using a surrogate biomarker, a surrogate endpoint, which means basically you can get an early approval and you can confirm post-approval that you actually have a clinically meaningful, durable benefit. But the point is it gets to patients earlier based on clinical trials. And so we need to keep this framework. It's very important for patients to get access to these therapies. There are a few other things that are important as well for our industry. There's a priority review voucher that is an important program. It helps fund innovation in this field, which you have heard is greatly needed to continue to treat patients, particularly in the rare disease field. And another thing that has been great, that is really helpful in our field, is the platform technology designation that has been put in place a few years back. And that framework allows to determine elements that you can use for several products that use the same technology. So in the case of what Dr. Musunuru explained, for example, if he wants to treat other patients with the N-of-1 approach, he can perhaps identify building blocks in the development of that product that can be reused. And so what this platform approach allows is that when you do that, you use some similar elements across different products for different patients, you can limit the amount of studies that you have to do. So that time frame of 10 years that I talked about can be shrunk. And it's important to know that the baby KJ who was treated, the approval was under a compassionate use approval. It is not an approval that would allow treatment of a large number of patients who would have different mutations. So this platform approach creates the framework to allow that. So that's great. We've made a lot of progress. We need to continue to build on that. So looking further, what else can we do, right? Because that duration of getting these products approved is still very, very long. So we want to accelerate cures. And I think there are some good ideas out there. One of them is, I think, accelerating the back and forth with the agency. Right now, if you ask for a meeting with the Food and Drug Administration, it takes about 2 months to get an answer. That's great when you can be forward-looking and you can actually wait for these answers. But what if you have an urgent question that's gating in your development and maybe it's just 1 or 2 questions? So we need to make sure that they have enough staff to be able to answer these questions, and we need to increase the informal back and forth to be able to resolve these issues. I think that's really, really important. There's a lot of discussions around AI and novel technologies. I think AI can help triage what is sent to the agency so that they can decide, is it really novel? Is it high risk? Does it need review? Or is it something that's pretty standard? We can have a quick review and we're done, or send a quick answer to the sponsors. And then I think eventually these gene therapies are going to benefit patients the most before they actually have symptoms or irreversible sequelae. So I think the agency also needs to think about what does the preventive treatment of patients look like. And then there's novel science now where gene therapy is used in utero, and that will create additional consideration on the regulatory framework. So in a nutshell, the Office of Therapeutic Products within CBER has done a fantastic job in supporting this innovation, but there is a lot more to do. So we look forward to partnering with them.
That's great. So you framed a lot within the approval process that's happening right now and needs to happen. And Erica, you're on the Hill a lot. You can probably dive a little bit more on the access side of it. What needs to happen in Medicare and Medicaid just to make sure these patients can actually access once the approvals happen?
Yeah, so Dr. Musunuru so eloquently articulated the criticality of NIH funding and support, and AV obviously, talking a lot about how the FDA and the regulatory process can impact ultimately the development of treatments and the timeline for that. So I'm going to talk a little bit about CMS, the Centers for Medicare and Medicaid Services. I'm biased. I worked at CMS for a number of years on Medicaid policy. But there's a couple of things. There's a piece of legislation that the Alliance for Regenerative Medicine and IGT have long been advocating for. It's called the Accelerating Kids Access to Care Act. It's a bipartisan bill. Last year, we had over 200 co-sponsors in the House. We had almost half of the Senate co-sponsoring the bill. It's something that actually, up until this weekend, was part of the reconciliation legislation, and we're hoping that there's still a possibility that that could pass either with reconciliation or this year. And what the Accelerating Kids Access to Care Act, as the name implies, is it does, in fact, accelerate kids' access to care. One of the barriers that kids face, and particularly kids like Andrew and like Callipy or MLD patients, is that they have to travel. Nate mentioned they had to go from Chicago to Columbus.
40 times.
40 times.
Yeah.
And Maria, you mentioned, not only these patients traveling, and the first patient treated in the U.S. Traveled all the way from Alaska to Philadelphia a number of times. And Maria, you highlighted the huge number of clinicians and providers that were involved in this specialized care. And so what happens is each state, each Medicaid program has to actually credential all of the providers that are taking care of a child. So if you have a child from Illinois who's covered by Illinois Medicaid, and you are receiving care at Children's Nationwide, all of those providers at Children's Nationwide, any providers that maybe exist if they went to CHOP or another center of excellence, they all need to be credentialed within the state of Illinois' Medicaid program, or in the case Maria described, in the state of Alaska's Medicaid program. And those Medicaid programs have different resources, they have different products, processes. This is very wonky, but it's a great example of sort of the need to alleviate administrative burden. I think that's been a hot topic on the Hill for a while, another bipartisan topic. It's also a big focus of this administration, sort of alleviating administrative burden, taking care of things that stand in the way of patient care. And so the Accelerating Kids Access to Care Act basically streamlines that process. It creates sort of a streamlined enrollment that makes it easier. That's going to take less time for those clinicians. There have been a number of stories in the news about patients who have had to wait, and as Nate articulated, time is muscle, time is functionality, time is ultimately life or death. And so if we can get rid of those administrative delays, that would make a huge impact. It also alleviates some of the burden on providers. And Dr. Musunuru, I'm sure you and your colleagues can attest, we want you in the clinic, we want you treating patients, we don't want your sort of clinical staff and administrative staff expending resources on all this back and forth. And I know UPenn and CHOP in particular, they treat patients from all over the country. I've talked to folks at CHOP about this. So Accelerating Kids Access to Care Act, please, let's get that done. Another thing that AV mentioned that's really important is the accelerated approval pathway at the FDA. So you talked about the importance of that, the importance of being able to approve therapies based on surrogate endpoints. It's not conditional approval, it's full FDA approval. And what we've seen, unfortunately, is we've seen some insurers and we've seen state Medicaid programs basically sort of taking another look and saying, we don't think accelerated approval is full approval. It's experimental. We're not sure we're going to cover it in the case of commercial insurance, or we're not going to cover it in the first year of approval. We've seen state Medicaid programs say, well, it's not fully approved, so we're going to pay less. We're going to pay less for that drug than if it were full approval. Or we're going to restrict coverage. We're not going to cover it to label. We're going to only cover it for the clinical trial population that's smaller. And so that's a problem. That's a big problem, because that is ultimately undermining the purpose of accelerated approval, which is to help patients with high unmet medical need. And it's harmful to patients. And so one of the things that we need to do is we need CMS to hold those state Medicaid agencies accountable to make sure that they are fulfilling their statutory obligation to cover FDA-approved drugs. And we really need the agency to sort of put that pressure on states to ideally put out some guidance, but really hold them accountable and their managed care plans accountable. And that's just a really critical thing for these patients. And then finally, I'll just, Avi did a great job of talking about it, but you talked about, the progress at OTP. Obviously, we have new leadership at CBER. Folks are getting to know Dr. Vinay Prasad, the new head of CBER. He's made some comments about sort of the importance of pushing therapies for rare disease, which is great to hear. But we want him to continue that sort of forward-looking momentum that we've seen at OTP and under Dr. Nicole Verdun. And you mentioned things like platform technologies, which is a great example of how guidance is actually helpful for bringing cures. So we know this administration is really focused on alleviating administrative burden. They want to sort of find ways to reduce the amount of regulations, which I think we all want to get rid of red tape, but in some cases guidance is helpful and necessary, and we need to preserve that. And so what that means for you on the Hill is that we just need to make sure that we're not throwing the baby out with the bathwater, that the agency is sort of continuing to provide those guardrails and that guidance that all the stakeholders need. And Avi, as you intimated here, we need to make sure that our federal agencies have the funding that they need, but also that they have the staff that they need. So when we're talking about things like the reorganization of HHS or the reduction in the federal workforce, we need to make sure that we're not interrupting critical functions, and that's not just reviewers, but the policy folks doing this important guidance. And we want to make sure that they have the right expertise. And unfortunately, reductions in force and attrition, that can sort of slow down, as you mentioned, the approval time or make them less responsive. You're not going to have, more frequent phone calls when you have less staff with higher review loads. And so I think that's really important. And then the last thing I'll just say is obviously strong collaboration with industry experts is really important. We saw these FDA CEO roundtables. I hope there'll be some meaningful follow-up from that. And then I would love to see Dr. McCary did these CEO roundtables, and there's a few more happening, but we'd like to see patient roundtables as well, because that voice is critical. And historically, FDA and NIH have had a rare disease day. They've brought people in. That was postponed. And so I would love to see the FDA do some patient roundtables to complement the CEO roundtables. So I'm sorry, I think I went over time, but a lot of things. And the bottom line, some of that stuff I mentioned is stuff that we'd like the administration to do. So accelerating kids' access to CARE Act. Let's make sure we have robust appropriations for the NIH and the FDA. And then also, as you're working with these agencies, either exercising appropriate oversight, asking questions, reaching out. Hearings are great. We'd love to get these guys up, not only to brief you, but we'd love to have them testify. I think they've all auditioned. They all would make great committee witnesses. So with that, I will turn it back to you, Eric.
That's a great highlight of policy initiatives to focus on and pay attention to. But I think if I look at Maria and Nate, maybe you can just comment real quickly also. What would you want policymakers to think about, to understand, as families are navigating these rare or life-threatening conditions, to make sure that they're going to be able to be treated? Preventative medicine, treated with cell and gene therapies. What do you think policymakers need to be thinking about?
So I've got my Illinois commercial driver's license. As I mentioned, I work for a family-owned trucking company. And my father-in-law, upon hiring me, insisted I get a Class A license. And I was pretty ticked about that, and I was pretty scared about that, because Chicago is a pretty nasty place to drive an 18-wheel tractor. But eventually I finally did pass the test. So the analogy I want to give is this. When you're in a tractor trailer, 18-wheeler, it's a manual transmission, and you've got a 40,000-pound load, and you're facing an uphill climb, and you have to come to a stop because of a traffic light or someone's going to turn. If you take your foot off the clutch, you start rolling backwards. And we can't have that happen here in America today with gene therapy and CRISPR. We've got to mash the gas pedal down and run it up to about 4,500 RPM and keep going forward. So,
Perfect.
There you have it.
There you go. Maria.
I want to answer that question with the fact that when I get a phone call from a parent and we have a baby that needs treatment, I have to tell those parents, we have no time for you to cry. I need to get you on a plane to one of the 2 centers in the country that can treat your baby. And if you know a U.S. senator or a CEO at your insurance company, you might want to get them on speed dial. And honestly, when I deal with a family, the first thing I ask them is, what state do you live in? Because that's going to have profound implications of whether they're going to get reimbursed in time. and the docs can't see them unless Medicaid signs off. So I've had families wait months to see a QTC specialist, and these babies need to be treated in 6 months. And my families from certain states where there have been massive delays have not enjoyed the incredibly transformative power of this therapy because it came too late. And we need to make sure that where you live and who you know and how much money your family has and who is in your phone does not depend on, will not impact if your child gets to live. And right now, the good news is every single child who's needed Lenmeldy has gotten it, but every single time they have gotten treatment, I have had to convene a war room, work with the press, and call U.S. senators. To get Nicholas from Alaska to Philadelphia required the entire Senate delegation of Alaska. Now, I thank the Alaska Senate delegation for their incredible work to get Nicholas there, the fastest baby we ever got to CHOP, actually. But it should not take 2 U.S. senators to get a baby a treatment.
Perfect. Well, that kind of leads in, maybe we can have each panelist give 1 minute as we're rounding out to close here, but just maybe 1 minute, what's at stake if the United States fails to modernize our healthcare or our regulatory system and keeping up pace with this innovation that's out there?
Well, I think that's actually really easy because it started happening at some point in gene therapy when regulations were not evolving quickly enough. What happens is companies go and start their clinical trial outside of the US. And so you've seen companies in the past 4 or 5 years start their clinical trials in New Zealand, Australia, and other areas in the world where the approval to initiate a clinical trial, that first milestone I talked about, is more straightforward and has a clearer and faster process. So I think the impact is if our regulatory framework doesn't continue to support innovation in this field that's going fast, and we want to go even faster, we're going to see innovation go elsewhere.
Yeah, I mean, we're starting to see glimmers of hope where there wasn't hope before. And so, think of it as a door. It's slightly ajar, light is starting to shine through. And it's really this country more than anyone else in the world who has been responsible for, kicking it slightly open. And we need to kick that door wide open. And it should be us. We started this. We need to be the ones to finish it. I'll leave it at that.
Great. Erica, too.
I mean, I mentioned obviously some things that Congress can do, but I mean, what's at stake, AV? You're absolutely right. The U.S. is going to sort of lose its lead in terms of innovation, but ultimately patients, like Nate's son, like Maria's daughter, and the patients she helps, they're not going to have access to care. Patients could die, and that is horrible. And so I think, we need to make sure that our federal policies and all of you can continue to support these agencies, can continue to put forward innovative, forward-looking regulations that keep pace with science, that we are investing in these not only these incredible cures, but Dr. Musunuru, you pointed out the importance of foundational research. We have heard some sound bites in the news and some folks in Congress say, well, look at this silly study that the NIH is funding. Bubbles, why are we studying bubbles? Why are we studying yogurt? Well, because that is ultimately what led to CRISPR gene therapy and treating babies like KJ. And so we need to invest not only in the development of these treatments and clinical trials, but in that kind of foundational research. And Dr. Musunuru, I'm sure you can attest to one of the things that I worry about too is not just companies taking their trials elsewhere, but I worry about researchers going elsewhere, not coming to the US. We have a lot of researchers and PhD students who are international. We have researchers and physicians here who are going overseas. And so that brain drain isn't just going to jeopardize America's sort of leadership in the biotech innovation, but it's going to mean fewer treatments. It's going to mean restricted access. You're not going to see sort of the same robust capacity at centers of excellence like CHOP, like Nationwide Children's, like Children's National here in DC. And we need to keep those folks here. That's really important for patients. It's really important for science. And I guess I'll stop because I really want to make sure folks have time for questions.
Good. Nate, anything to wrap up?
No.
Anything else, Maria? Okay.
I think we covered it.
No, I just think that after the the work of these amazing doctors, heroic, brilliant doctors, is under a real threat. Without NIH funding, no one is going to invest in high-risk research like this. And no one's going to invest in research which will not have a commercial application for a pharmaceutical company. So Dr. Aaron's and Dr. Musunuru's work wouldn't be possible without the NIH. And we want more KJs. We don't want KJ to be the last baby that is saved this way.
Let's have a round of applause just quick for our panelists. All right. I do want to thank all of you for taking the time. Thanks for taking the time to join today. Please know also that you can reach out to ARM, to IGT, if we can be a resource to you, if we can be helpful. Visit our websites at gene-therapies.org or alliancerm.org to learn more. We do look forward to being a resource and interacting with you in any way possible. But also, we'll close now, but feel free to engage in our panelists offline if you like and ask them questions. So thanks again.